Hello everyone, I Dr. Sharmishta Banerjee welcome you all on the SWAM course in Genetic Engineering and its recent advancement. Today we are going to discuss microarrays. Till now we have studied the fundamentals of genetic engineering, different types of vectors, libraries and their screening strategies, gene expression and its regulation in prokaryotes. So let's moving on to the today's topic, that is microarrays. Before moving ahead, I would like to add why we are studying this topic or what are the potentials of microarrays. Microarrays chipping away at the mysteries of science and medicine. Microarrays are a powerful tool for measuring gene expression. Now the question is, what is gene expression? It is a term which is used to describe the transcription and the information contained within the DNA. Scientists study the details and kinds and amount of mRNA which are produced by a cell to learn which genes are expressed, which in turn provides insights into how the cell responds to its changing needs. Microarrays are revolutionizing medicine by being able to pinpoint a very specific disease or the susceptibility to it. Before moving into the details of what are microarrays, I would like to discuss history of microarrays. This landmark discovery began in the year 1991. Scientists at the California-based biotech company Affymax produced the first DNA chips. Affymetrix, a company dedicated to gene chip technology, was established in 1993, and in the same year it created many microchips. Today the company has a variety of gene chips, including ones which can identify the different HIV strains or can detect mutation in cancer genes. In 1995, researchers at Stanford University find a way to make DNA chips in the laboratory. In 1996, researchers put DNA chips to the test for the very first time. In 1996, Francis Collin uses an Affymetrix microarray to detect mutation in the breast cancer gene, that is BRCA1, breast cancer associated gene 1. It is basically a group of genes which are responsible for causing breast cancer or are associated in the development of breast cancer in women at risk for the disease. Within a year, Brown and his colleagues created the first chip containing all 6000 yeast genes. In 1998, researchers began to use DNA chips to identify polymorphisms. In 1999, DNA chips proved valuable for classifying and studying cancer. Now the question is, what are microarrays? It is basically a compact device that contains a large number of well-defined immobilized capture molecules. These captured molecules can be synthetic oligos or oligonucleotides, PCR products, proteins, antibodies which are assembled in an addressable format. I will discuss what do I mean by addressable format in later slides. You can expose an unknown or a test substance on it and then examine where the molecules are captured. You can then derive information on identity and amount of captured molecule. Microarrays, they are also known as DNA microarrays or gene expression microarrays. They are powerful tool in molecular biology and genomics which are used to analyze the expression levels of thousands of genes simultaneously at a single point of time. They allow researchers to investigate how genes are turned on or off in response to various conditions, diseases or treatments. Here are some of the key aspects of microarrays. Microarray. Now the question is, what is a microarray? From the name itself, what we can understand? Array. It is basically a combination of rows and columns. So these microarrays are a miniature form of an array, we can say, that we can see here in the figure, that it is a combination of rows and arrows, but it is miniaturized, or since it is miniaturized, we call it as a lab on a chip or biochips. It is basically a semiconductor device. It's used to detect the DNA makeup of a human cell. Now, we all know that microarrays contains millions of DNA strands which are designed to pair with their other half as the liquefied human cells are poured over them. This principle is basically the hybridization process, and this can be further detected with the help of a laser technology. The principle of microarray, it's again very simple. The whole principle revolves around principle of hybridization. Now, the question is, what is hybridization? We all know that when two things hybridizes, when two things they overlap over one another or they stick over one another, that is known as hybridization. So, the main principle is hybridization. It involves binding of the complementary DNA or the RNA sequence. In a typical microarray experiment, a glass slide or a silicon chip is spotted or synthesized with thousands of known DNA or RNA probes which are very specific to a particular gene. Now, the whole microarray, it is basically a combination of rows and columns. Either it can be a 96 microtiter plate or it can be a multiple of 8, any rows and columns which are multiple of 8. The lowest microarray chip which is available is 96 microarray plate. So, what is basically happening here? The chip is immobilized with a certain type of probes. What are these probes? They are basically single stranded oligonucleotides which are labeled with some sort of technologies. They can be fluorescent labeled, they can be luminescent labeled, or they can be radiographed. So, or they can be labeled with the help of a radioactive molecule. So, these are the probes. These probes are immobilized and the complementary DNA or RNA sequences will be present in the sample to be tested. So, in this way, when we find the complementarity between the test DNA or the RNA sequences as well as the probe, there will be hybridization, and these hybridization signals are then detected by the help of the mechanism by which the probes were labeled. So, in total this is how the microarrays they work. If microarrays are so good, there is a question, why didn't we use them before? So, there are the reasons for that. Now, we will discuss one by one in brief. Not all of the genes were available at that particular point of time. No SNPs were known, that is no marker single nucleotide polymorphism were identified before. No suitable bioinformatics tools were available for the analysis purpose. New proteins are now becoming readily available nowadays in comparison to the conventional times. So, these are the reasons why they are not used before. Microarrays and the associated technologies should be regarded as the byproducts of the genome initiative, nanotechnology and bioinformatics. Microarray analysis stage, it can be divided into three main steps. The first one is array fabrication or probe preparation. What is array? I had already discussed. Now, we will discuss one by one all of the steps in details in further slides. The second step is sample preparation and hybridization. The third is data collection, normalization, analysis and its interpretation. This is the summarized form of the whole DNA microarray experiment. The first step is prepare your DNA chip using a chosen target DNA or RNA. Generate a hybridization solution containing a mixture of fluorescently labeled cDNAs. cDNAs are basically complementary DNA. They are synthesized from mRNA by using reverse transcriptase. So, what is the benefit of using this cDNAs? That only the expressed parts are basically sequenced in the form of cDNAs through mRNA. So, we need not study the whole genome to study the differential gene regulation in different organism or in different species, or to compare a cell from a healthy one to distinguish it from a diseased individual or diseased cell, we can say. So, cDNAs are more beneficial rather than using the complete genomic DNA for gene expression studies. The third step is incubating this hybridization mixture containing fluorescently labeled cDNAs with your DNA chip, that is basically your probe. The fourth step is detect the bound cDNA using laser technology and store the data in a computer. And the last and the very important or the foremost step is analyzing these data using computational methods or bioinformatics tool. Now, how do we prepare the sample? The first step is sample preparation. The target DNA or RNA is often labeled with a fluorescent dye and is extracted from cells or tissues of interest. The target is then applied to microarray chip where it hybridizes with the complementary probes. The degree of hybridization will indicate the abundance of specific mRNA or DNA in the sample. That means, if there is higher degree of hybridization, the intensity of the signal which it will produce or the intensity of the fluorescence which it will produce will be more. So, we all have seen the first figure that there were small dots, red, yellow, green dots. So, the color, the red color, if it is more dominant, that will indicate the degree of hybridization is more. So, based on the amount of the DNA or the mRNA which is present in a particular sample, the color intensity will change and it will indirectly give us the correlation of degree of hybridization. Second step is data collection. Microarray scanners are used to detect the fluorescent signals which are emitted by the hybridized targets. The intensity of these signals corresponds to the expression level of the genes represented by the probes. Software is then used to analyze and process the data. The question is how the array is fabricated. The array fabrication is done in again two different steps. First is probe preparation and second is the arraying of these probes with a specific address onto the chip. So, how the probes can be prepared? So, the probes can be cDNA, it can be a PCR product or it can be oligonucleotides, that means they are the completely synthesized nucleotides in lab. So, these probes, what is the basic identification factor or what is the basic characteristics of these probes are, it must be complementary to the sequence of gene of interest or to the sequence which is under study. So, it can be prepared in many weld, multiple weld micro titer plates. Second is the arraying of these probes moved from micro titer to glass slide by a robotic arrays. They are basically denatured, they are basically deliberately made single stranded. How? By heat treatment or by exposing them to heat. So, this is very fundamental or important step, I will say. Denaturation is mainly important because if these probes are not denatured, they will not bind or they will not hybridize to our test molecule or to the molecule of our interest. So, these denaturation is to be done to compulsorily convert the probes into a single stranded, and it is very easily done by exposing them to heat or higher temperature. The third step in probe arraying is drying it. In this lithography means the light is used for writing, produced using photolithographic technique. So, this I will discuss in the later slide. Lithography, from the word itself, it means writing on a stone. Photolithography, what does it mean? Writing on a stone with the help of light or with the help of photons, packet of light we can say. So, the probes are basically captured or embedded on these chip, on these microarray by this particular technique, photolithography. This process is similar how we make the semiconductor chips. All of us, many of us have seen a television IC chip. in the same way the microarray chip also looks like. So, the silver color small IC is basically embedded, and how it is embedded? It is embedded with the help of lithographic technique or photolithographic technique. mRNA samples are processed simultaneously or separately instead of in pairs in case of Affymetrix microarrays. This is the principle of DNA microarrays or printing the oligonucleotides by photolithography. So, what we can see, there is a substrate on which in presence of a light there are different phenomena which occurs. So, these probes, they will be deposited on the substrate with the help of photolithography. As we can see in the first step, that in presence of light there is a masking effect. So, this masking effect is resulting in chemical coupling. So, we can see the difference that the probes are getting attached to the substrate molecule. So, there is a lamp, there are two plates, one is for masking, and with the help of the masking effect the probes are getting embedded or they are getting immobilized on the chip that we can see in the later half of the figure, in the B part of the figure. This is how Affymetrix labeling is done. It is almost similar to the normal labeling techniques, but since we are dealing with Affymetrix microarray, so here is an outline of the same. Detection mechanism, again it is very important. So, what is detection of or what is detected here? Hybridization detection is performed using anti-digoxygenin monoanticlonal body which is labeled with alkaline phosphatase. So, many words are here, I will focus one by one. Anti-digoxygenin monoclonal antibodies. So, anti-digoxygenin, digoxygenin is basically a chemiluminescence molecule. Here we can say the blue color is indicated with the help of digoxygenin, and the red color is chemiluminescence molecule, the black color Y shaped compound is monoclonal antibodies, the blue color, light blue or greenish color is basically your alkaline phosphatase, and the test compound is basically, or the cell is basically your yellow in color. So, what is happening here? Anti-digoxygenin monoanticlonal bodies. So, this is a dual hybridization detection technique, like one by one each of them are complementary to another. So, what we can see, if the chip is having a particular gene of interest, then it will get attached to the monoclonal antibodies which are labeled with alkaline phosphatase. So, here the attachment has taken place, but the chemiluminescent molecule will only attach where the gene of interest has hybridized with this digoxygenin label monoclonal antibodies. That is why the red color compound is your actually anti-digoxygenin. So, the digoxygenin is basically complementary in structure to the anti-digoxygenin molecule. I hope I am able to make it clear. So, when these red molecule will hybridize, then only the signals can be detected or the result analysis can be done or positive signals are obtained, we can say. So, when there is a hybridization with the gene of interest, then only this type of chemiluminescence. Chemiluminescence means the luminescence in presence of certain chemicals. There will be chemical reaction which will lead to the luminescence phenomena, and this chemiluminescence signal is then actually captured or acquired by a CCD camera. Hybridization detection here in this particular figure, it is performed using antibiotin monoclonal antibody labeled with cytosine 3. The principle will remain same, but the molecules are different. So, here the detection mechanism of fluorescence signal is with the help of a laser based scanner. Hybridization is actually pairing. So, in detail already I had discussed what is hybridization. This figure will make you a more clearer concept of the principle of microarrays. The human DNA sample which has been replicated millions of times and fragmented into short pieces is washed over the microarray. The red balls, they depict, the red balls in the figure, they will depict the biotin molecules that were adhered to the fragment, and it swims around the probes for up to 16 hours in search of their complementarity sequences. During that time, some strands will pair with the probes. That process will be known as hybridization, whereas some will be left intact. Now, when the washing solution will be used, all the unwanted probes will be removed off. Only the hybridized pairs will remain there at the site. The next step is to detect these pairs with the help of some signal mechanism. The array is rinsed and washed with a fluorescent stain that clings to the biotin on the strands of the human sample that remain. A laser causes them to glow, and the DNA is genotyped or analyzed based on which the probes on the array they mated with or they hybridized with. Protein microarrays are lagging behind DNA microarrays. Same idea, but immobilized elements will be protein here instead of nucleic acid. Antibodies for high density microarrays have limitations because they are cross reactive to one another. Aptamers, these are basically the RNA molecules which bind efficiently to the proteins. They are known as aptamers, or engineered antibodies or proteins may be the viable alternatives for it. These are the applications of microarrays. Gene expression profiling. Microarrays are widely used to compare gene expression pattern between two different samples, that is healthy as well as disease, or control versus treatment groups, which will help the researcher to identify the genes associated with any specific condition. The next one is genotyping. They can be used for genotyping by detecting SNP or other genetic variation. They can even detect copy number variation, that is they can detect alteration in DNA copy numbers, such as if there is any gene duplications, deletions, insertions, or any. Epigenetics, they are used in epigenetic studies to access the DNA methylation patterns. MicroRNA profiling. Microarrays can also be designed to analyze high expression of the microarrays, small RNA molecules involved in gene regulation. Environmental conditions play an important role in gene regulation. A comparison of microarray data may reveal genes that are induced under one set of conditions and repressed under other. Genes that encode protein participate in a common metabolic pathway and are often expressed in a parallel manner. This can be revealed from a microarray analysis. The application overlaps with the study of gene regulation via microarrays. It can be used for screening of small molecule targets, post-translational modification, protein-protein interaction, protein-DNA interaction, enzyme assays, as well as epitope mapping. Advantages: it is a high throughput technique, it can simultaneously analyze thousands of genes, it has versatility, they can be applied to various types of biological samples, it has the characteristic of data reproducibility. There are certain challenges: limited dynamic range, cross hybridization, limited genome coverage. While microarrays were once a dominant technology in genomics, next generation sequencing techniques such as RNA sequences have become increasingly popular due to the ability to provide more comprehensive and quantitative gene expression data. But nevertheless, the microarrays still have utility in certain research applications and they remain a valuable tool in molecular biology. These are the problems which I had already discussed, and we can see in the left hand side the miracle of data analysis, microarray data analysis. This is a quick recap of already what we have studied in flow form. So, thank you for patience hearing, and in the next lecture we are going to study what is DNA footprinting, knockout technologies and much more. So, looking forward for this lecture. Thank you.